everyone. So today in this video, we would like to explain on the fundamentals and process of producing a glow-in-the-dark mice. However, before going any further into the video, we need to ask ourselves this. Why do scientists bother in producing transgenic animals, such as the aforementioned glow-in-the-dark mice? Well, one of the reasons is to help in the study of a certain gene's function and expression. Next, it helps in the process to procure a cure for incurable diseases. And finally, it can help to produce gene products, the likes of which can act as therapeutics. Moving on, the main components in this process are crucial to the success of the overall idea. The main components include the plasmid as a vector, the GFP coding sequence as a DNA fragment intended to be inserted, and E. coli acting as the host. Now, the first step on the process to create a glow-in-the-dark mice is to cut the DNA sequence encoding for insulin gene expression with the help of EcoR1, which is a restriction enzyme. This EcoR1 will help to create sticky ends, which increases cloning efficiency.
For step 2, after DNA fragment of the insulin gene was cut by the restriction enzyme, the green fluorescent protein GFP genes was inserted into the cleavage insulin gene sequence using ligase enzyme to undergo ligation, then bind the insulin gene sequence and GFP gene sequence. On step 3, single inserted gene that consists of the binding insulin gene and GFP gene will undergo ligation and will be ligated into a vector plasmid to produce a recombinant DNA molecule. One of the genes that is inserted in plasmid is antibiotic resistant gene. The vector will transport the recombinant DNA of the GFP and insulin gene into the cell, which is the virus, in petri dish that contain colonies of bacteria. Step 5. The virus that contains the vector recombinant DNA of GFP and insulin gene is then released into a male pronucleus of fertilized oocyte and will add the recombinant DNA of insulin and GFP genes into each cell. Next, fertilization occurs. Step 6. Within the cell, the vector multiplies, producing numerous identical copies not only of itself but also the gene that it carries that produce the integration of zygote genome and recombinant DNA. Step 7. Inject or implant the zygote containing desired recombinant DNA into the mouse. Step 8. Then, the cell inside the mice body will divide and copies of the recombinant DNA molecule are passed to the progeny and further vector replication takes place. Step 9, or the final step. All the founders should be screened for the presence of the transgenes to establish separate transgenic line.
After the whole process is finished, we still need to identify any potential founders. To do so, we first place the mice under UV ray to help regulate the transgene. Next, we obtain the genomic DNA either via an ear punch or tail snipping. Then, we need to screen for the genome presence. One of the methods is by using PCR, in which the obtained blood sample is added with other components into a thermal cycler. This one cycle process begins with the denaturation of the DNA strand before proceeding with the annealing and the extension of said strand. Another screening method is the Southern blot method, which although it seems a little more complex, is actually highly recommended due to the low false positive rate.